Good morning, everyone. I hope you were able to take a look at the link that I posted for the molecular biology review session. Since we have gone through the molecular biology part, now we are going to move on to the recombinant DNA technology. And once again, if you have any questions, feel free to either email me, come to the WebEx office hours, or post your questions at the Stellar Forum, and we'll be happy to respond to you in a prompt fashion. Again, for this module, we are going to look at the problems, and as we look at the problems, we are going to go through the major concepts that were covered in this module. So let us get started. Let us say you are looking at the pathway that leads to the synthesis of amino acid leucine in the yeast. Remember, yeast is a eukaryotic cell. When you look at this hypothetical pathway in the yeast, you realize that here A is converted to B, B to C, and C to leucine. This reaction is catalyzed by enzyme E1, which is encoded by gene E1. B to C conversion is catalyzed by enzyme E2, encoded by gene E2, and C to leucine is catalyzed by enzyme E3, once again encoded by gene E3. You're working with two different forms of the yeast. One is the wild type yeast, and this yeast is capable of synthesizing leucine because it has in gene E1, gene E2, gene E3, which are producing the normal functioning E1, E2, and E3 enzymes as shown by the plus signs over here. As a result, the wild type yeast is capable of synthesizing the amino acid leucine, and therefore it is able to grow in a medium independent of the presence or absence of leucine. On the other hand, you also have the mutant yeast. Just like wild type, the mutant yeast has normal E1 gene and E2 gene. So it is making normally functioning E1 enzyme and E2 enzymes, which are capable of catalyzing the conversion of A to B and B to C. However, it has a non-functional, it has a mutation in gene E3, as a result of which it is either not making enzyme E3 or is making a non-functional enzyme E3. As a result, it cannot catalyze the conversion of substrate C to leucine amino acid residue. Hence, this mutant is not able to grow in a medium that lacks leucine. It is dependent on the availability of leucine from an external source in order to grow, divide, and make more of its kind. So, what is your objective? Your objective is you want to clone the wild type version of gene E3 into a plasmid, a, a vector, and you want to use this recombinant plasmid to revert the mutant yeast to the wild type yeast. So the question is, how should you proceed? So your first step is you start with the wild type yeast. As we know, its genotype is E1 plus representing the wild type phenotype, E2 plus and E3 plus. You isolate the genome from this wild type yeast. Now, this genome, here's just a representation of the genome, and this genome will have the recognition sites for restriction enzyme X at different locations. So you cut this genome with the restriction enzyme X, which makes a cut between G and T over here and G and T over here. Similarly, G and T over here, G and T over here, G and T over here, and G and T over here. So when you cut the yeast genome with restriction enzyme X, you will end up getting multiple genomic fragments. These fragments will be of different sizes in terms of the base pairs because genes come in different sizes. However, since you are generating these genomic fragments by using only one restriction enzyme X, so all of them will have the sequence TCGACG at this end and the sequence GCAGCT on the other end. So let's say this is the genomic fragment representing gene E1. This is the one representing gene E2. Similarly, you'll have a genomic fragment for gene E3, 4, 5, 6, and so on and so forth. So this should be your first step. Starting with the wild type yeast, cutting the genome with a restriction enzyme to get multiple genomic fragments, all of which will have the same sequence at their ends, but all of which these will be of different sizes in terms of the number of base pairs because genes come in different sizes. So as a next step, your step 2 is you take a plasmid. So what is a plasmid? From our lectures, we know that a plasmid is a self-replicating circular DNA. And why it is self-replicating? In specific species, because it should have the ORI site specific for those species. So let us say you are using this particular plasmid. So this plasmid has the yeast ORI site as well as the bacterial ORI site, which means that it is capable of replicating within the yeast as well as in the bacteria. It also has a selection marker over here, which is the AMPR gene. So this gene, when it is expressed, it makes a protein or an enzyme which can actually degrade the antibiotic ampicillin. Now, along with ampicillin as a selection marker, it also has a screening marker, which is the LAG-C gene. This gene, when expressed, makes an enzyme which can convert the substrate white XCal into a blue color product. So any bacterial cell that has an intact LAG-Z gene will appear blue when it is grown in the medium that contains the substrate for LAG-Z or which contains XCal, which is a substrate for LAG-Z. So you start with this plasmid and you already know the features of this plasmid. Now you digest this plasmid with the restriction enzyme Y, which is able to recognize this particular sequence. It's important to know that the sequence generated by by cutting with restriction enzyme Y is complementary to the sequence generated when you cut the genomic DNA with restriction enzyme X. You will see that in a minute, but just bear with me for a moment. So you digest the plasmid with restriction enzyme Y, and when you do that, you get this digested plasmid. So it has all the features: yeast ORI, bacterial ORI, AMPR, degraded or inactive LAG-Z gene because you are cutting it with restriction enzyme Y. So you have disrupted the LAG-Z gene, and at this end it will have CGAGCT as the end sequence, and at this end it will have TCGAGC over here as the end sequence. Now, so you have digested the plasmid and you have digested the yeast genome with two different restriction enzymes, restriction enzyme Y and restriction enzyme X. So you bring them together and you add the ligase enzyme. So since the ends generated by digestion with restriction enzyme X and Y are complementary, they will base pair with each other and the sequence will be made continuous by the action of DNA ligase enzyme. So let's say this is a recombinant plasmid where the insert over here or the cloned insert over here is gene E1. Similarly, you will be getting another recombinant plasmid that has gene E2, gene E3, gene E4, and so on. So you'll get many such recombinant plasmids, each containing one specific yeast genomic fragment as an insert. So together, you call this the ligation mix. So next what you do is you take the bacterial strain. We need to remember that our original plasmid had the AMP resistant gene. And it also had an intact LAG-C gene. So you should be starting with the bacterial strains that are sensitive to ampicillin and those which lack the LAG-C gene. So you take the AMP sensitive LAG-C minus bacterial strain and you transform these bacteria with the ligation mix that you have prepared. When you do that, you end up generating 3 different populations of bacterial cells. Bulk of the bacterial cell populations will remain untransformed, so they will not take any plasmid whatsoever. They'll neither be taking the original plasmid that had no insert, nor will they be taking the recombinant plasmid that should be having one of the yeast genomic DNA as the insert. So these untransformed bacterial cells will continue to stay AMP sensitive and they will be LAG-C minus. So they will not be growing in the medium that lacks they'll die in the medium that contains ampicillin. I need to correct this. So these plasmids will die in the medium that contains ampicillin. I'm going to correct this. The second population will be the transformed bacterial cells, but these bacterial cells will be the ones that will be transformed with the original plasmid. So they will continue to be they will become AMP resistant because they are receiving the AMP-R gene as a part of the plasmid, and they will also be LAG-C plus. So they will grow in a medium that contains ampicillin since they are able to degrade ampicillin. And since they have an intact LAG-C, they will be able to convert the X-cal substrate into a blue color precipitate. So they'll grow and form colonies all in the ampicillin containing medium, all of which will appear blue. The third population will be the transformed bacterial cells that will have the recombinant plasmid with with different yeast genomic inserts. So they will have an intact AMP-R gene, but they'll have a disrupted LAG-C gene because you are putting the yeast genomic fragment at into the plasmid by disrupting the LAG-C gene. You're putting it at the Y site. As a result, these bacterial cells will be able to grow in a medium that contains ampicillin, but they will not be able to convert X-cal into a blue color precipitate. So they will form colonies in the medium that has ampicillin, but these colonies will continue to remain white in color. It is these colonies, the colonies growing in the medium that contains ampicillin and the ones that appear white, it is these colonies that you are interested in because together these colonies have the entire information of the yeast genome in them. So what you have essentially done is you have constructed a wild type yeast genomic library in the bacterial cells. Great. But next, what you need to do is you're not interested in all of the genes at the moment. All you are interested in is the yeast gene E3. So, how can you fish out that gene from the yeast genomic library that you have prepared within the bacteria? So, you can do so if by a technique which is called colony hybridization, and you can use this technique if you have some information about the sequence of gene E3, which you are interested in over here. So let us say this is a part of, this is a partial DNA sequence of gene E3. So if you know this sequence, you can use it in order to design a probe, a DNA probe, whose 5' end is radiolabeled. Okay, so you take, so here's the plate that has the bacterial colonies. So, and these bacterial colonies together are representing the yeast genomic DNA library within them. You take this plate, you put a piece of paper on the plate, and you take the impression of all these colonies, keeping the orientation the same. After that, you degrade the bacterial cells. But when you degrade the bacterial cells, the DNA that was in the bacterial cells of that colony will remain stuck exactly at the same point. Now you take your radiolabeled probe, you add it to the paper, and you incubate it for a certain time interval. After this, you take the paper, you put an X-ray film on the paper, and you develop it. Let's say this is the colony that lights up. If this colony lights up, then basically you know that your radiolabeled probe has hybridized to the complementary sequence of complementary sequence, and this complementary sequence is basically the E3 gene. So your radiolabeled probe over here has hybridized to the complementary sequence on the E3 gene, which, and this E3 gene was a part of the recombinant plasmid present within this colony. And that's why it is only this colony that is lighting up. So you started with many, many colonies, but by doing the colony hybridization, you have fished out the colony that has the recombinant plasmid with E3 gene as the insert. Okay, so now what you do is you isolate the plasmid from this colony, and you so you have isolated the plasmid from this colony, and then you take your mutant yeast and you transform the mutant yeast with the plasmid, recombinant plasmid that you have isolated. After transformation, you grow the mutant yeast in a medium, in the growth medium that lacks the amino acid leucine. Any colony that grows on the growth medium that lacks the amino acid leucine is the colony that is now capable of synthesizing leucine. So it is basically it is a representative of the colonies that have been converted from the mutant which were initially the mutant yeasts, but now have been converted into the wild type yeast. So all the colonies growing on a plate that has growth medium lacking leucine represent the reverted colonies, the colonies that have reverted from the mutant yeast phenotype to a wild type phenotype. So you have successfully converted the mutant yeast to the wild type yeast by inserting a recombinant plasmid that has the wild type form of the E3 gene as an insert. So your first experiment is successful. So now let's move on to the next experiment. So before we do so, let's take a look at this slide. So this slide is basically representing representing what we just went through. Yeast cells isolate the chromosomal DNA, degrade it, and then basically clone it. And so what you have done is the mutant yeast had a defect, had a mutation, as a result of which they were not able to synthesize amino acid leucine. Since you are inserting the wild type copy of the E3 gene, so you're complementing the mutation. And therefore, this experiment is oftentimes referred to as cloning by complementation. Now let's move on to the next experiment or the next problem. Let's say you are very much encouraged by your first the results of the first experiment, and now what you want to do is you want to clone a human gene in the bacterial cells. Not only do you want to clone the gene, but at the same time you also want to express the gene. So this is a little tricky because although almost all of our cells have the same genome, different cells express different sets of genes, and therefore they have different mature mRNA pools. So let's say if you want to clone the insulin gene, although the insulin gene will be present in all cell types, but you will start only with with the pancreatic beta cells, because it is these cell types that express the insulin gene to make the insulin protein. So when you are trying to clone and express a human gene, you have to start with a specific cell type that is expressing that gene, and hence is having the mature mRNA corresponding to that gene. Secondly, you should not be cloning the whole gene, but as I said, you should be starting with the mature mRNA. Because if you want to clone and express the gene, a human gene, in bacteria, you need to remember the bacterial cells do not have the splicing machinery. So when you're looking at a gene, it has the introns and it has the exons. So if you are cloning a gene and are trying to express it in bacteria, the introns will not be spliced out. So you will not be getting the protein. So instead of cloning the whole gene, what you do is you go to a specific cell type and isolate the entire mature mRNA population from that cell type. So over here, as an example, if you want to clone and express the insulin gene, you start with the pancreatic beta cells that secrete insulin. And if you want, yeah, so you start with these. So we know that the mature mRNA that you have isolated, so you have isolated the mature mRNA pool from a specific cell type. All the mature mRNA population will have a 5' cap, but along with that, they will also have a 3' poly A tail. You are going to exploit this feature because since there is a 3' poly A tail, all you can do is you can come up with an oligo DT primer, which will undergo complementary base pairing with this 3' poly A tail, as is shown over here. So the base pairing is through the hydrogen bonds, and the primer is anti-parallel to the mature mRNA. So 3' end is facing the 5' end. Then you use an enzyme called reverse transcriptase, which can basically read the mRNA 3' to 5' to make a complementary DNA strand, or the first strand 5' to 3' by elongating the 3' hydroxyl end of the last T in this oligo DT primer. So you got your first strand of DNA. Now you don't care about the mature mRNA, so you can simply degrade it. So you have the first strand of DNA, and now you can use this as a template with the help of DNA polymerase enzyme to make the second strand of DNA. So what you are generating is a population of cDNAs, each of which corresponding to a specific mature mRNA type in the mRNA population that you have isolated from a specific cell type, in this case the pancreatic beta cells. So here your starting point is a specific cell type, and you're using the mature mRNA population to make a cDNA populations as we described over here. Now, step 3. So you have the cDNAs, And now you can, so let's say this is the sequence that is recognized by restriction enzyme X, and this is recognized by restriction enzyme Y. I'm just giving these as the hypothetical enzymes over here. So you can clone this in a particular plasmid by using the DNA ligase as we had described earlier. So let's say the plasmid that we are using over here is essentially this plasmid. This plasmid has the bacterial ori, so it should be able to self-replicate in bacteria. Once again, it has AMP-R as the selection marker, and it also has the LAC-Z gene as a screening marker. But along with this, it has the site for restriction enzyme X and restriction enzyme Y. The difference between this plasmid vector and the plasmid vector that we used in problem 1 is this plasmid vector is containing a bacterial promoter. And this is very critical. We need to remember that we made the cDNAs by using the mature mRNA. So the mature mRNA is essentially only the exons. It lacks the introns, and it also lacks the regulatory regions of the genes such as the promoter sequence. So if you are cloning a cDNA in the plasmid and you want to express the cDNA, you have to clone it. You have to have the right promoter, and you have to clone it in a way so that it is oriented correctly with respect to the promoter. Okay, so let's say you cloned it in this particular plasmid. So once you clone it, again you do the selection and screening by using the ampicillin in the growth medium, and also by providing XCAL. Any colony that grows in the ampicillin containing medium and appears white due to a disrupted LAC-Z gene is actually containing one cDNA insert in the as a part of the recombinant plasmid. So what you have essentially done is you have created a cDNA library in the bacterial cells. So again, you have so once you have created the cDNA library, you have to figure out which of the colonies in this bacterial colonies in this cDNA library essentially has the gene of your interest. So you can once again do a colony hybridization to figure out the bacterial colony that has the gene of your interest. So I'm going to diverge a little bit over here. Let's say this is the insulin gene. And for the sake of this experiment, let's assume that it has the site for restriction enzyme Z, and it also has a site for restriction enzyme A. So you are getting this insulin cDNA as a Z cut fragment. And you clone this in this plasmid that has the site for restriction enzyme Z as well. The distance between the recognition sites for restriction enzyme Z and A is 1 kb. The plasmid as a whole is 4 kb, which means that the sequence from A all the way up to here is 3 kb. So once you clone it, there are two possible ways by which this insulin cDNA can be cloned over here. It can go this way as shown over here. So Z, A, Z, with this being 1 kb, this being 2 kb, and this being the direction of transcription. Or it can orient in the reverse way as shown over here. If you cut this plasmid with restriction enzyme, if you isolate this plasmid, let's say from bacterial colony A, and you cut it with restriction enzyme A, then you will end up getting two fragments. One will be of the size of 2 + 1, 3 kb, and the other will be of the size 3 + 1, 4 kb. If you get the fragments of this size, you know that this is the orientation of your cDNA insert in the plasmid. and this is the right orientation with respect to the bacterial promoter. So the colony that has the recombinant plasmid with the cDNA in this orientation will be expressing the cDNA, and you will be getting the protein corresponding to that cDNA. But if you take a look at this one, you cut it with restriction enzyme A, you get 1 plus 1, 2 kb, and 3 plus 2, 5 kb. If you get these fragments, you know that your insert has been oriented in a direction which is opposite to the promoter. So although you do have colony, bacterial colony B, which has the right insert, right insulin cDNA insert, this cDNA will not be expressed. So it's actually of no use to you. So not only do you want to insert the cDNA into the plasmid, but you want to insert it in a way that is correct with respect to the promoter so that it can be expressed. Okay. So once again, this is shown over here, and basically I'm repeating the same point over here. Colony A has the insulin cDNA insert in the right orientation with respect to the bacterial promoter. So this colony will express the cDNA and make the insulin polypeptide chain. Okay, so yes, you did make a human protein in the bacterial cell. So you use the bacterial cells as the factory to make the human protein. Now you're even more inspired and you talk about your experiments with a friend of yours. And your friend says that she wants to clone yet another human gene, BAL1 gene. She wants to make the BAL1 cDNA, so she isolates the BAL1 cDNA from the cDNA library that you have prepared. And but she wants to use a different approach. She wants to make it as a fusion cDNA. She wants to fuse the BAL1 cDNA, this being the direction of transcription. She wants to fuse the end of the BAL1 cDNA with the beginning of the green fluorescence protein cDNA. So she wants to make a BAL1 GFP fusion cDNA as shown over here, with this being the direction of transcription. So what are the precautions that she should be adopting? First of all, when she is fusing these two, she should be absolutely sure that she wants to maintain the reading frame of the BAL1 cDNA as well as the GFP cDNA. Since she wants to make a BAL1 GFP fusion cDNA, which can be expressed to make a BAL1 GFP fusion protein, she absolutely needs to delete the stop codon over here. Otherwise, the GFP will not be expressed. But she wants to maintain the stop codon of the GFP. So these are the precautions that she needs to take. Now, this slide should be very familiar to you because I have this as a part of recitation 9 for you, and you must have practiced this already as a part of your recitation handout in your recitations. So let's say this is the BAL1 cDNA and the stop codon is underlined for you. You use this information to figure out the reading frame of this cDNA. Similarly, the start codon for GFP is underlined for you. So you use this information to figure out what is the right reading frame of the GFP. Both of these have the recognition sites for restriction enzymes Z and Y. So you want to figure out which of these restriction enzymes should you use in order to make the fusion cDNA. If you do the question on your own, you'll see that you'll use the restriction enzyme Z, not Y, because it is this restriction enzyme that will maintain the reading frame of BAL1 as well as the GFP cDNA. It will delete the stop codon that is at the end of BAL1 cDNA. But if you use the restriction enzyme Y, you are changing the reading frame of the GFP cDNA. So this will not give rise to the fusion protein. So yes, you did make the cDNA. Now what you want to do is, okay, I do have the cDNA, but I want to get it in bulk amount. So you want to amplify the cDNA. And you do this with the help of a polymerase chain reaction. So let's say this is the beginning and the end of the fusion cDNA. You use this information to design primers. So first of all, let's say this is your fusion cDNA. You subject it to a high temperature so that the two complementary strands separate from one another since the hydrogen bonds between the complementary bases are broken. And then you reduce the temperature from 95 to, let's say, 56 degrees centigrade. You add the primers. So let's say this should be the forward primer, which is complementary to the bottom strand, and its 3' hydroxyl end can be elongated to make the top strand. And this is the reverse primer, which is complementary to the top strand, and its 3' hydroxyl end can be elongated to make the bottom strand. So you lower the temperature, add the primer, and let the primer undergo complementary base pairing. Let the primer undergo annealing. After that, you increase the temperature once again to 72 degrees centigrade. So at this point, a thermostable DNA polymerase called Taq DNA polymerase will elongate the 3' hydroxyl ends of the forward and the reverse primer. So starting with one DNA molecule, at the end of one PCR cycle, you'll get two DNA molecules. You go back and you repeat the cycle. So at the end of second cycle, you'll get 4 DNA molecules. Third cycle, you'll get 8 DNA molecules, and the process can be repeated. So if you repeat the cycle n number of times, at the end of n cycles, you will get 2 to the power n number of DNA molecules. So this is a very good technique to amplify the DNA. So you amplified your PCR, your fusion cDNA using PCR, but now you want to make sure that nothing went wrong during the PCR amplification. In order to do so, you do a DNA sequencing reaction. So for this, this is the general procedure. For the original Sanger DNA sequencing, you do the reaction in 4 tubes. Each of the tubes should have just one DNA template. Remember, in PCR you had 2 DNA templates. Here, all you need is just one DNA template. You add this to each and every tube. You have to have a DNA primer that is complementary to a 20 to 25 nucleotides long sequence on the DNA template. The primer undergoes complementary base pairing with the DNA template, and it is anti-parallel as well. So you add the primer to all of them. You absolutely need to add DNA polymerase so that it can elongate the 3' hydroxyl end of the primer. You add all the DNTPs, A, T, G, and Cs, to each and every tube, 1 through 4. But the difference between the tubes is with respect to the dideoxy NTPs, which lack a hydroxyl group at their 3' position. So let's say tube 1 has DDA, tube 2 has DDT, tube 3 has DDC, and tube 4 has DDG. So you let the reaction proceed, and then you get DNA fragments of different sizes, and you resolve these fragments on a DNA sequencing gel. So let's say this is the sequencing gel that you end up obtaining. Now the fragments, we know that DNA is negatively charged, so it will be moving from negative terminus to the positive terminus when you apply the current. And the smallest fragment, which will be the closest to the 5' end, this will be the fragment that was synthesized first. So, and this being the smallest fragment will move faster compared to the other one. So the fragments move from negative to positive terminus, but when you're reading it from bottom to top, because this fragment will be the closest to the 5' end, and this will be the closest to the 3' end. So if I'm reading it, I'm going to read it as 5 prime, C, A, G, C, and so on. And ultimately, this would be C, A, 3 prime. So smaller fragments will be at the end of the sequencing gel. You read the gel from bottom to top to get the DNA sequence 5 prime to 3 prime, and the template DNA sequence will actually be complementary and parallel to the sequence that you just obtained. So, well, you got your fusion cDNA, you PCR amplified it, you checked its sequence to make sure there was no mutation, and you did this by DNA sequencing, original Sanger sequencing method. Nowadays, people we don't we try not to use the radioactivity. So the modified version is, instead of using radio labeled dideoxybases, you can just use fluorescent tag dideoxybases. And so you don't need to do the reaction in 4 tubes. You can just do it in 1 tube. You can run a capillary gel, and then basically a detector and a computer program will give the sequence to you. So this is the fluorescence dideoxy sequencing method. Okay, so let's say here's your BAL GFP fusion cDNA. This is the sequence at the end, at the beginning, and this is the sequence at the end, and you ligate it in a plasmid over here. This is a slight diversion from what we have been discussing so far. Here I'm using two enzymes which are generating ends that are not compatible. So remember, in question 2, we looked at two possible orientations because we were using restriction enzyme, the same restriction enzyme, to cut the plasmid as well as the cDNA fragments. But here I'm using two different enzymes which are generating ends that are not compatible. So there are not two possible orientations. There is only one possible orientation of your fusion cDNA in the plasmid. So if you use this approach, that allows you to do directional cloning, and by doing so you can make sure this is the only way that the insert goes into the plasmid, and if it goes this way, it is in the right orientation with respect to the promoter, and it will be expressed. Okay, so these are the three problems that I have with respect to the cloning. Now let's move real fast to SNPs, which is a topic that was discussed in Recombinant DNA Lecture 4. Well, we know that we have approximately 20,000 different genes, and our genome is about 1 billion base pairs long. And if I sequence my genome and compare it with the genome sequence of any of you, you'll find that 99.9% of our genome is just the same. But still, we are so different from each other. We have different heights, eye color. Some of us are left-handed, some are right-handed, and so on and so forth. These differences are because of single nucleotide polymorphisms, single base pair changes, which are distributed all across our genomes. These single base pair changes are referred to as SNPs. I would strongly recommend that you take a look at this site. I think this is also posted as a part of the lecture slides. This is a very good link to understand what SNPs really are. So let's say you have one individual has this sequence as a part of the gene, and another individual has this sequence. In another individual, the corresponding sequence is this. So this individual, you can say that this individual has AT as a SNP or GC as a but individual 2 has GC as a SNP. Oftentimes we say that individual A has A as a SNP, individual 2 has G as a SNP instead of saying AT SNP or GC SNP. Now, SNPs, these single base pair changes, they happen at they are seen at a frequency of more than 1% in the general population, whereas mutations happen at a frequency of less than 1%. So they are different from mutations. Now they are seen at a frequency of 1 every 300 base pairs. So basically, across our genome, we may have these many SNPs, and that accounts for the differences that we see amongst us. Most of the SNPs are non-consequential, and they can be seen within the gene or regions between two genes or in the regulatory regions. But the good point about SNP is they can be used as markers to predict different traits, different diseases, and so on and so forth. So let's take a look at an example. Let us say you are looking at gene D, and this can exist in the form of two different alleles, allele big D and allele small d. You look at a large cell population, large population, and you see that if individuals for the if an individual has SNP AT, or basically just the top strands A as a SNP at this position, the individual has allele big D. But if an individual has T at the same at this position, the individual has allele small d. So A and so if an individual inherits this chromosome from the dad and this chromosome from the mom, then the profile, the SNP profile of that individual is A, comma T. Or you can also say the SNP profile of that individual is T, comma A. So keeping this in mind, let's take a look at this pedigree. What's the mode of inheritance? From your lectures in genetics, you know that, and also from the SNP profile, you can see that the males over here only have one SNP. So clearly, this is an X-linked recessive mode of inheritance. The people who have T as the SNP are the ones that are shaded. So T is the SNP that is associated with this particular trait or this particular disease. So SNPs are often used to predict the mode of inheritance of a disease. And they're used for forensics to find out criminals, and they have many uses. And I have some practice problems which at the end of this presentation, which you can take a look at and solve on your own. Okay, how can you detect SNPs? The easiest is you can simply sequence the genome, but you can also do so by doing a SNP microarray. This is a small chip of about 1 centimeter square in size, and it has thousands of these spots. Each spot over here represents multiple copies of oligonucleotides, which are about 20 to 25 bases long, and each oligonucleotide has a specific SNP. Now, if you want to identify the SNP profile of an individual, you take the genome, fragment it, denature the fragments, and put it on the SNP chip. If, let's say, this oligonucleotide has a T as a SNP over here, and a genomic fragment from an individual binds to this oligonucleotide, you can say that the SNP profile of that individual at this position is A, because T will base pair with A. So SNP microarrays are often used to identify the entire SNP profile of individuals. Now, real quick, we also looked at gene editing, and I would strongly recommend that you look at the slides that are posted by Professor Siv, and she has voiced over those slides, so they are very, very beneficial. So she used over here in this particular part, we are talking about CRISPR, using CRISPR through gene editing. Here what you do is This is a gene editing method which was originally found in bacteria, and the bacteria use it as a defense mechanism against viruses. But now we are exploiting it to edit our other genomes. So here, the first step is you come up with a guide RNA, which is shown in green. This undergoes complementary base pairing with one of the two strands of the target site, and it does so left to the 5' end of a sequence called PAM site. Once there is complementary base pairing, the DNA duplex at the target site denatures or unwinds. Following this, the Cas9 endonuclease comes and cuts the two strands. So, this is the cut that is shown. The repair enzymes will try to repair it, but in the process of repairing it, it will generate point mutations which can change the frame. So let's say if you're looking at a disease where the affected individual have the genotype big A, small a, essentially the disease is a dominant disease. So if you can use the CRISPR-Cas9 complex and you can target the dominant allele, then basically you can provide relief to that particular individual. That's the whole idea. Now, finally, in the last few minutes, we are looking at how can we generate transgenic animals. Let's say this is one phenomena. Here you take the fertilized eggs. So these eggs should have a specific genotype. Let's say it's big A, small a, small a. You change the genetic makeup of the fertilized egg by introducing an allele of a specific gene. So, since all the cells in the individual arise from the in this mouse will arise from the fertilized ovum, all of them will have this introduced gene or introduced transgene, and um, so so you implant this into a pseudopregnant female, let them develop into the pups, and all pups are transgenic because they are expressing the gene that was introduced into the fertilized egg. So this is an example of transgenesis by injection. Once again, please take a look at the slides and the voice and the commentary posted by Professor Sev. Another example is transgenesis by chimera. Here you take an early embryo. Let's say this is the recipient embryo. You take cells from a donor embryo as well. You implant these cells into the recipient embryo. You insert the changed embryo into a pseudopregnant female and let it develop into the pups over here. So the cells that will be originating from these inserted green cells will likely have the genotype different from the cells of the original embryo, which are shown in purple. So in the same mouse, you can have cells which will be you can have a mixed cell population. Some cells having one genotype if they are coming from these green cells, and other cells having yet another genotype if they are coming from the original purple cells. So this is an example of transgenesis by chimera. So this is a brief introduction of all the most of the points, if not all, that we went through in the recombinant DNA module. I would recommend that you take a look at these practice questions, do them on your own, and if you have any question, please let me know. Thank you, guys.